Thank you so much, everybody, for joining the webinar series, The Use of New Approach Methodologies for the Risk Assessment of Pesticides. This series is being co-organized by PETA India, the Biological Agri Solutions Association of India, and the Society for Alternatives to Animal Experiments in India. My name is Ankita Pandey, and I'm an advisor to PETA India, and I will be co-moderating today's webinar with Mr. Vipin Saini from the Biological Agri Solutions Association of India. Today, we will hear from Ms. Pritee Singh from J Research Foundation, who will be speaking on the use of RTgill-W1 Cell Line Assay to assess acute fish toxicity. Before I introduce the speaker, I would like to point out that there will be time for questions for the speaker at the end of the presentation. Everyone is on mute, but you can type your questions or comments in the question and answer box at any time during the presentation. This toolbar should be just below your screen. So, Ms. Pritee completed her Master of Science degree from Maharaja Sayaji Rao University of Baroda, Vadodara in 2017. She is presently serving as the study director at J Research Foundation, specializing in aquatic and terrestrial studies. In this capacity, she is responsible for designing experimental protocols, study initiation, comprehensive data analysis, and interpretation. Beyond these core responsibilities, Ms. Pritee plays a pivotal role in conducting validation studies, detailed study reports, ensuring regulatory compliance that contributes to advancing scientific knowledge and supporting environmental safety. It is now my pleasure to turn the floor over to Ms. Pritee.
Thank you, ma'am, for introducing me. So, I will begin with my presentation. Good afternoon, all. So, use of RTgill-W1 Cell Line Assay to assess fish acute toxicity. My screen is visible.
Yes, Pritee. Yes.
Okay. So, moving toward the presentation, that is environmental risk assessment. So, ERA is a process of identifying, evaluating, and then mitigating the potential harm to environment. So, conducting this environmental risk assessment of a chemical is one of the important steps for ensuring the safe use and management. This plays a vital role in regulatory framework like classifying, labeling, and packaging of a chemicals. In aquatic toxicology studies, fish are the important, or we can say the key indicator for this water quality assessment and contaminations due to the sensitivity to the substances. For registration of a new or any conventional pesticide, regulatory framework requires may require acute toxicity data, either one on cold freshwater fish, one for warm freshwater fish, or one saltwater fish. So we can see here how many number of fishes are utilized for a registration of a single pesticide. The traditional aquatic toxicology study rely mostly on in vivo testing where the whole animal was utilized. Such approaches often significantly require the resources and raise the ethical concern over the use of live animals. Fish cell line has emerged a new approach methodology for environmental risk assessment or aquatic studies. So, alternative to fish acute toxicology, uh, there are two guidelines. That is, one is OECD 236 fish acute embryo toxicity test, and the second one is fish cell line acute toxicity test. That is by using the RTgill-W1 Cell Line Assay. For OECD 249, we will be discussing in the upcoming slides. In OECD 236, that is a fish embryo acute toxicity test, is a replacement of OECD 203, where juvenile and adult fish were utilized. In OECD 236, that is Along with the lethal concentration, the EC50 value is determined based on the mortality and behavioral symptoms. Along with this, the chemicals which are having the effect of teratotoxicity, or we can say having the effect on development of an embryo. So such chemicals can be evaluated with the help of this embryo acute toxicity test. We all know that zebrafish is having the development of zebrafish can be observed with our naked eyes and can be recorded under, or pictures can be captured by using the microscope. So here, further chemicals which are having teratogenic effect can be easily identified with the apart from the lethal and sublethal effect or EC50 value. The other important point that we can gain from OECD 236 is developmental deformities. The studies are up to 96 hours and extended sometime up to 120 hours. Ethical, if we will see this study, then biologically it is a in vivo study, but ethically is a in vitro study in some countries. But this means alternative for OECD 203 is accepted in some countries, and some are again against this proposal. Uh, in country like Brazil, still it is protected under animal law. They are considering it as an animal, but ethically it is said that if an embryo is feeding or taking the nutrition from their yolk, it is not considered as an animal till after 120 hour they are capable of feeding from the external source. So after 120 hour they will be counted as an animal, but under up to 120 hour they are not counted as an animal. So, alternative to fish acute toxicity test. In traditional fish acute toxicity test, a large number of fish were utilized. For testing at JRF also, we have conducted around 600 to 700 number of studies. In each phase, these studies for apart from knowing the some data about it, as we all know that there are published data available for particular technical chemicals, but for formulated there is no data available. So to determine the more accurate LC50 value, here we are performing the study in two phases. One that is a preliminary range finding study, and then the second one is main study or definitive test. So for a single phase, that is a DRF preliminary range finding study, also called as a DRF dose range finding. So wide range of concentrations are selected, and based on this concentration, main study is performed. So in single phase, that is a DRF or main study, around 60 to 70 number of fish are utilized for for a single phase. Then counting for both phases, around 120 to 140 number of fish are utilized. Apart from that, the number of fish, the quantity of test item. Here, a large quantity of test item are utilized. In fish acute toxicity study, the test solution is prepared based on the body weight and length of the fish. So, according to the species, there is some variation. So, around 40 to 40 liter of test solution are prepared. So, this large volume of test solution is prepared. Accordingly, the quantity of test item is also requested or requested. Then the duration of study. Here for a single phase, the study is up to 96 hours. And then for another phase, again it is of 96 hours. So long duration, around 2 weeks are gone for a testing a single chemical. And the end point is to killing the sorry. The end point is killing the we have to kill the fish if it is not dead. Then here the important thing that is a species wise, we have to keep a dedicated room also. So for a single species, a single room is prepared. A single room is dedicated for a single test. Apart from this all, is there any question? Apart from all these things, the large number of tanks are also utilized, um, and the of different size. The end point is to determine the LC50 value or 100% mortality. If data available, then NOAC or LOC. In revised guideline, there is no requirement for NOEC and LOEC. So OECD 249 has brought a very means good guideline, that is an alternative. From a single fish, the gill tissues are extracted, and from this gill tissue, gill cells are extracted and cultured in a 70 cell culture flask. Here for guideline has recommended two subculture flasks, that is 75 centimeters square and 25 centimeters square. These cells are immortal cells that can multiply forever. And instead of tank, we use here as a 24 well plate. In this 24 well plate, again the volume of test solution is reduced. Apart from 4 to 40 liter, we can use here 2 ml of test solution. Then a tank size is also reduced, and more parameters are detected from this test guideline. With the less quantity of test item, the duration of test is of only 24 hours, and the end points are, and we have to calculate here NOEC, LOEC values. In such a way, the concentration has to be selected. Now, refinement to fish acute toxicity test. This is in contrast to environmental risk assessment. So, OECD has validated many guidelines in section 2, that is a effect of uh, living organism with the effect of chemical or effect of toxin with the living biotic system. So here I have segregated some guideline, that is OECD 210 fish early life stage toxicity test. In fish, the guideline itself says that is a early life stage toxicity test. So here the early life stage of fish are exposed to different concentration of test chemical. Most probably the exposure is within 2.5 hours of post fertilization. So this on the basis of the result of this toxicity test, the lethal and sublethal effect are calculated, and EC50 value is also calculated. EC10, 20 and 50 value is calculated along with NOEC and LOEC value. So the data collected from this guideline will be important for assessing the chronic toxicity of a chemical. Now OECD 212, that is a fish short-term toxicity test on embryo and sac fry stage. This test we can say as a preliminary screening for long-term toxicity test. Here, the studies is till until the embryo consume or absorb its nutrient. So, on the basis of this, we can select a concentration for long-term toxicity test. Now comes OECD guideline 215, that is a fish juvenile growth test. As we all know that juvenile is a period where growth and development of an organism occur. So here the fish is exposed, or the embryos are exposed at the juvenile stage of their development. Fish are exposed at the juvenile stage of development. So growth rate, the toxicity is assessed based on the parameters like their growth rate and body weight. In OECD test guideline 229, that is a fish short-term reproduction assay. This fish short-term reproduction assay try to mimic the aquatic environment where sexually mature male and female fish are exposed to the endocrine disrupting chemicals. So this fish, sexually mature male and female fish, how they are getting affected with these chemicals, and the effect are observed on their upcoming generation along with them also. So the adult fish, their vitalogenin content, here the parameter is detected with the content of vitalogenin, and then on the secondary sexual characteristic. This is one of the guidelines where the effect can be observed on the upcoming generation also. That is in detail, that is a gene can be that is a we can affect the parent animals and the gene level of upcoming generation. In the upcoming generation, how the genes are getting affected and how the development is also getting affected with these endocrine disrupting chemicals. So for a long term assay means for having a long research, this guideline is one of the best guidelines. Then OECD 234, that is a fish sexual development test. Here, newly fertilized embryos are exposed to the endocrine chemicals which affect the sex determination of a chemical, sex determination of the embryo. So, based on the vitalogenin content and secondary sexual characteristic or gonadal histopathology, the fish sexual development or the toxicity can be assessed. Then in vitro method, continuation with the refinement to fish acute toxicity test. In vitro methods that support the 3R. These guidelines, there are some guidelines that have been mentioned in section 3, that is environmental fate and metabolism. So I have mentioned here the three guidelines. One, that is a 319A. In 319A, that is a determination of in vitro intrinsic clearance using cryopreserved rainbow trout hepatocytes. So here, suspension of enzymatically active cryopreserved hepatocyte or hepatic cells are exposed to the test chemicals. Based, then samples are the reactions is stopped at a specific time interval, and then by adding a stopping solutions and with the validated analytical method, this decline or decrease in concentrations are measured. Then on this basis, intrinsic clearance is calculated, and then it is how it is means it is important for predicting the toxicity of a chemical. Similarly, in 319B, liver subcellular fraction is used, where this subcellular fraction contains a cytosol and microsome enzymes. So, these microsomal enzymes and cytosol metabolize the chemical, parent chemical, and again here also at different time points the sample is collected and intrinsic clearance is calculated, and on the basis of this the the toxicity is predicted for that particular chemical. In OECD 305, that is a bioaccumulation in fish, aqueous, and dietary exposure. So here the fish are exposed to the test chemicals by aqueous and dietary exposure. There are two phases. One, that is the uptake phase, and the second one, in uptake phase, the chemicals are exposed, and then in second phase, that is a depuration phase, where they are then given means we can say recovery phase, where they are again kept in the clean and non toxic chemical, means clean water, and after that lipid content is collected from there, and based on the lipid, the bioaccumulation or the chemical, how much it get accumulated in the tissues are identified or determined. Now moving toward the guideline, that is a OECD 249. So RTgill-W1 cell line was initiated from gill filament of a healthy rainbow trout. So rainbow trout is one of the standard species in many guidelines along with OECD 203. These gills are the unique organ for gas exchange, osmoregulation, and many other critical functions that is required for the survival of fish. So gills are the primary target and uptake site for water contamination. RTgill-W1 cell line are monolayer and are adherent to the subculture flask. They are growing attached to the subculture flask and are single layer. The cell line is propagated in subculture flask at 19 degree without additional supply of CO2. The medium which used here is L15 medium. So the medium has a physiology to maintain the pH. So there is no need of additional CO2 supply. At low density, the cells are irregular in shape and are irregular in spindle shape. At high density, after reaching the confluency, they are tightly packed and are polygonal in shape. So application of this guideline supports to generate the toxicity data. Along with that, it is important in QSAR relationship, means QSAR, that is a quantitative structure activity relationship, which predict the chemical behavior based on their molecular structure. And then on weight of evidence approach, where all available informations are important for assessing the chemical toxicity. Its first application in context to environmental risk assessment was for effluent testing, then application for ISO guideline adopted in 2019. These cells are important for important model for in vitro aquatic toxicology as well as for gill function and gill disease, for studying the gill function and gill disease. The cells has been used to understand the mechanism of toxicity, potency of toxicity, and evaluating the toxicity of environmental samples. The principle of study is RTgill-W1 cell line in a confluent monolayer are exposed to different concentration of test chemical in 24 well plate for 24 hours. Then cell viability is assessed after 24 hours with a set of fluorescent cell viability indicator dyes. And the end point is to determine the EC50 value, NOEC and LOEC. In this continuation with this principle, three different dye has been used here. One that is a risazurin based dye. The second one is CFDA and third one is neutral red. So risazurin assess the mitochondrial or membrane activity. Then CFDMA assess the integrity of a cell membrane and neutral red assess the integrity of a lysosome. So these are the based on the these three dye fluorescence of these three dye the results are determined. Risazurin enters into the cell in its non-fluorescent form and then it is converted into product called risorufin by mitochondrial, microsomal and cytoplasmic oxidative activity. A decline in risorufin indicates that there is some disturbance with the mitochondrial membrane or microsomal activities. CFDA enters into the cell, and if the cell has an intact membrane, then they get converted into fluorescent substance called carbofluorescence. But in absence of this substance, it is there is a no fluorescence, and there is some disruption or decline with the disturbance with the plasma membrane of a cell. Lysosomal, that is a neutral red. Neutral red enters into the cell and diffuses into the cell and then accumulates in the lysosome. So a decline in neutral red fluorescence indicate that there is some disruption with the lysosome membrane. The material and method for validation at JRF, we have validated this study, and at JRF we have used positive control, that is a 3,4-dichloroalanine. It is also recommended by the guideline. Then 3,4-dichloroalanine is an intermediate of pesticide, pharmaceuticals, and raw material for agriculture pesticide. It is easy to handle and is used as a positive control in zebrafish acute embryo toxicity test. The test system which we used here at JRF was RTgill-W1 Cell Line that has been procured from ATCC. Moving to our experimental procedure. So a single flask here, that is first day, that is a day one of the experiment. On day one, we perform the cell seeding. The cells are seeded in 24 well plates. So here a subculture flask of 10 to 12 days old that has reached the full confluency is utilized here. So a subculture flask is taken. The media in the media already present in the subculture flask is removed, and then it is washed with it has been washed with a washing for twice, and then as the cells is having the cells are having the property of adherent. So to detach the cells from the subculture flask, trypsin is used. So up to trypsin has been used. The concentration of trypsin was maintained 0.25%. And to stop the activity for few seconds, after few seconds of trypsinization, media was added. That is a L15 culture media to stop the activity of trypsin. Then this material has been transferred into a centrifuge tube, and after centrifuge, in the pallet, the fresh medium is added, and manual cell count has been performed with the use of hemocytometer and microscope. So 350,000 cells has been added in each well plate, in each wells in 24 well plate, except the first two. On day two of experiment, so here before preparing the stock solutions, it is mandatory from the guideline to observe the cell confluency of a cell. If the confluency is observed, then we can go for the treatment. But if the confluency is still remaining, then we have to keep it for another 24 hours. So here at JRF, we did 21 trials. Among these trials, on the next day, we obtained the confluency. So we prepared the stock solution immediately after the cell seeding. So here the stock solution as 3,4-dichloroalanine is insoluble in water. So a solvent, that is a DMSO, was utilized to prepare the stock solution. So in this, depending upon the concentration, the stock solution was prepared. As per the guideline, at 1%, the organic solvents are non-toxic to the cell, non-acutely toxic to the cells. but guideline has recommended to use only 0.5% volume by volume. So here we have prepared 200 times more concentrated stock solution and then added in L15 medium. So here in each well, 2 ml of stock solution was added. And then on day 3, cell viability was measured. In first two well, in first well, that is a A1 well, dosing mixture containing the highest test chemical was added. In A2, that is the second well, there L15 exposure medium is added. So to check the fluorescence of a test chemical, these two wells are kept cell free. The experimental design was we took there was six group. Each group was having a three replicate, and each well 350,000 cells were added. This was the concentration which we selected to validate the study, along with the negative control and solvent controls. Observation was up to 24 hours after exposure, and the endpoint was to determine the EC50 value. So these are the this was this based on their dye, the 2.5 standard deviation EC50 value is given in the guideline. Was given is still there in the guideline. So percent cell viability, we did 21 trials at GRF with different passage, and out of 21, we obtained in 13 we got the data or result as per the guideline. So for different time, this was the data which we obtained. In conclusion, the variation of raw fluorescence in two cell-free control, one with the L15 medium and the second with the highest concentration, the variation between them was less than 20%. The cell viability with negative control and solvent control was within the range of 10%. For 13 trials, we got these two data, and the EC50 value of for DCA in according to the dye, this was the data which was within 2.5 standard deviation from EC50 value recommended in OECD 249. Therefore, this study highlights the robustness of RTgill-W1 cell line and its accurate performance. And this can be used to assess the acute toxicity for fish in fish for pesticide and other environmental chemicals. This was the references which we have gone through. I have kept this slide here. 35 organic chemicals were used, out of which 73 were having the data with the difference of five-fold in vitro and in vivo study. The data of EC50 and LC50 value was having only difference of 35 five-folds. But in some chemicals which are having the property of hydrophobic, we're having the difference of 10-fold both in vivo and in vitro study. So RTgill-W1 Cell Line, apart from that, they have also mentioned that allyl alcohol chemicals were having the difference of 2,700 fold, which is not acceptable. And the chemicals which are having the, that is, we can say neurotoxic chemicals, which are based on the ion channels, sodium chloride ion channels. So they were also not giving a proper result, means around 190 fold or 63 fold differences for some chemicals they have mentioned here. So for neurotoxic chemicals and chemicals which are having the endocrine disrupting properties, this assay OECD 249 is not up to that mark. But for other chemicals, we can rely the data, we can compare the data, and we can say that they are accurate. So thank you all.
Thank you very much, Pritee. Wonderful presentation, quite an eye opener, I'll say. And thank you for sharing your insights. Pritee, you have a couple of questions if you'll be willing to attend to. I know you have to rush
So
post
it's
this
okay.
Zoom call. Yeah. So, Preeti, the first question that I would like to just place across that is this particular method, in vitro method, approved and used by the Indian regulatory authorities?
Yes, that's why in India it is means in India also it is getting validated.
Okay. And is it also good enough to test biologically based biopesticides, both botanical and microbials? Does it work for them? Have you conducted any studies on that?
So for microbial, I'm not sure because
Okay.
microbial is having some different properties. For a simple study, it is also
Yeah.
difficult.
Okay. Okay. And what about botanicals? It should be working over there, I believe.
Botanical also, sir. We
Okay.
are
Not not
talking.
not not not, not an issue. Not an issue. Okay. Coming to more technical question, how does this RT-Gill W1 cell line mimic the physiology of a live fish gill? Could you just elaborate on that question?
Sir, these cells that are derived from gill epithelium, and in live fish, gill epitheliums are the first site of taking these contaminants or water quality, water contaminants or any chemicals. So they have designed in such a way that gill epitheliums, they have extracted the gill epitheliums, which is a primary site for contaminants, taking the contaminants.
Okay, yeah, fair enough. And how does this TG249 results correlate with the LC50 values obtained under TG203?
Uh, that is also in restriction, as I said, that endocrine and neurotoxic chemicals, they won't work for 249, but other chemicals like means not 100% data is getting same data, but around this we can use as a pre-screening, means OECD 249, instead of, as I said, that in we are here not only here but in academic also and in other CRO also. To get an accurate value, they are performing two tests, that is a DRF and then main study. So based on 249, we can select the concentration, and whatever result here, with the help of three parameters, we will get the EC50 value. So the lowest EC50 value which we obtain in this OECD 249, that will be used as a concentration of uh, for this 203, means 203 uh, 96-hour LC50 value.
Okay, yeah, fair enough.
We can say this
And
as a preliminary screening for 203.
oh, okay. So it's a preliminary screening that we can bet upon, right?
Yes, instead of using too many animals, we can reduce the animal by preliminary
Fair enough.
screening this test.
Okay, that's great. We have another question popping up. It says, can TG249 be applied to testing complex mixtures?
Yes, it can be used by using OECD guidance document 23. We can go for this. We can use, but we have not till now. We have at JRF we have not conducted any study. But as per
Okay,
the guideline, they have said that we can use OECD guidance document 23, and based on that, we can at least try our test.
okay. Can you just elaborate further upon the scientific advantages of TG49, 249 compared to the traditional TG203 test?
Yes, one of the important thing is that ethically means large number of animal will be reduced, and then the duration of study can also be uh, means we can reduce the duration of study, the volume, quantity of test item, and everything. And the data which we obtain from 249 can be utilized. Means if we want to only check the preliminary screening or collect the data for environmental risk assessment, then this will be helpful.
Okay. There's one question coming up. Have you ever confronted other contamination during cell culture? And if you have, then how did you ensure that the cell line is free of contamination?
Actually, sir, we after several passage we are preparing, means collecting the samples and giving for microplasma test in different lab. So accordingly, based on their result, we can say our culture is free of contamination. Initially, when we have thawed one or two wells, then we found that there was some contamination, and with this means, the contamination was like fungal growth. So more precaution was taken thereafter, and then regularly we have sent our sample for mycoplasma test.
Okay. So, I mean, Just one other just query came over, probably maybe interest of the in the audience. How does TG249 fit into integrated approaches of testing and assessment, Ayata?
The data which we will be obtaining from here, along with other data also, that will be helpful for like
Take your time. We're in no rush. Just think it over. Not an issue.
means whatever data we will be getting for means whatever data, if I want to register for a new pesticide, then it will be required for me to collect the data of that particular test item. So this guideline will also help. Instead of using fish, we will use this guideline and we will perform the study, and based on this we will at least get some information of how the chemical is working, what is the molecular structure of this that particular chemical. So that will help in silico means designing all the other formulations or reducing the toxicity of that particular. So that will be important for production field, means those who are synthesizing the chemicals. We
Okay.
will collect data and we will submit them.
Okay. Just out of the thing you said that it is the TG249 is acceptable by the Indian authorities. Did you, prior to submission of your data, provide a comparative study of TG249 with the Vivo's studies, just to gain confidence of the regulators? Was any such kind of exercise being done?
No, sir. Actually, after validation, we were already busy with some other studies. So at JRF, till now, we have not done. We
Okay.
did only validation. No other
Okay,
chemical has been
but the
used.
studies you conducted for the Indian industry, for the to be submitted to the Indian authority, that was acceptable, if I may take it from you.
This we have validated at JRF, means we have validated, we are ready to perform the study, but till now we didn't receive any study from any regulatory, means any company.
Industry for the Indian Regulatory Authority. Okay, fair enough. Okay, so probably it will be nice because during the toxicological protocols being set over in 2017, this was one of the requirements. So probably to give JRF an edge, probably you could provide data generated under TG49 and compare it with a traditional in vivo acute fish toxicity and gain the confidence of the Indian regulators. I'm sure then most of the industry would like to do this. Nobody wants to kill more animals than than necessary, right? It's just a you can take it under an advisement. One thing I'll just say, you did mention that in Brazil. I mean, correct me if I'm wrong, if I'm wrong in my understanding. You did mention that in Brazil, it is considered as an animal, so it is protected under those laws. And in certain countries, beyond 120 hours, they are considered as animals, and in some countries, up to 120 hours, may not be considered as animals. So, I mean, how much is TG4249 internationally acceptable and validated in this regard?
Actually, sir, this acceptance is of 236 guideline, OECD 236.
Okay, okay. So
Biologically, it
not
has
to, so not up to 249, right?
no, no, sir, no.
Acha,
That is
and
a
any idea which global authorities are recognizing the use of TG249 in complete essence?
in EU. It has been accepted. European Commission, it has been accepted, but I'm not remembering, sir. I have to go through.
No, 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 not an issue, not an issue. It's just to give an idea so that we get to know. Okay, one probably this should interest you that how does TG249 compare in reproducibility and reliability to the traditional OECD TG203? fish acute toxicity?
Sir, here in 203, it is like fish acute toxicity. The any means at every time we are using the there is no reproducibility for that because the age, health, and everything size that will be differ. That will be different as we go for another test chemical or we perform. There will be difference in for a single chemical also. There will be difference in range finding and main study. At least some data are getting differ. But in OECD 249, from a single passage, from a single cell line, means it's single vial, we are preparing the passage. So same cells will be available. So there will be high chances of reproducible result.
Okay, yeah, fair enough. Uh, uh, Now this is just off the subject thing. I visited your institute in the late 1990s, and could you just elaborate, apart from this kind of study, what more in vitro studies that you conduct at JRF, if you could just elaborate, either on fish or otherwise? What kind of facilities, more about JRF, in fact, on in vitro or alternative studies? Since we have got some time, just a casual talk.
Sir, here in Ecotox, that is in Ecotox itself, we are trying to validate 319A and intrinsic clearance, and then 319B also. So these are the two guidelines for ecotox section. Apart from ecotox, in genotox and NAMS, many studies, in vitro studies has been validated, like skin sensitization. I'm not remember. Many
No, no,
studies.
it's all right. It's all right. It's all right. It was just a casual question. But thank you very much, Preeti. You've been such a resource to give us a more detailed, what I would say, insight into this kind of studies of TG 249 and others also. So thank you very much for all of this. And Ankita, I believe I'll hand it over to you.
Yeah, I know you can conclude. I'll just like to add here that,
Oh,
I think
yeah.
JRF has been quite active in, offering in vitro services like, Ecotalks. Of course, this is for the first time we touched upon Ecotalks, but in the last webinar series, we had speakers from JRF talking about in vitro, skin irritation,
Yeah.
eye irritation. We had one from QSRs. So I believe they're doing a lot of work in the alternative space. Just wanted to add
Yeah,
that.
thank you.
And
Thanks,
yeah.
Ankita. Ladies and gentlemen, this was Ankita Pandey from PETA, the Senior Scientist and Research Policy Advisor. And if you have any further questions, feel free to write her at the email shown on the screen. And Preeti, saying thank you very much and for your participation and taking out the time out of your, I understand from your busy schedule. I myself have had a stint as a toxicologist. I can understand how difficult it is to take out time from regular daily work. So thank you very much, ladies and gentlemen. It's been a pleasure of having all of you out over here, and the recording of this webinar will be available on YouTube and was already probably.
Thank you, sir.
I think we lost Mr. Seni. Okay, I'll just quickly conclude. So thank you so much, everybody, for your time. If you have any questions, please feel free to reach out to me. And you can email me at the ID given here. And just enjoy the rest of your day. Thank you so much, everybody. Thank you.
Thank you, ma'am.
Thank you.